Thank you all for joining the second session of Biomaterial Webinars. And today I'm going to present in vitro responses to biomaterials. So before starting or anything, I would like to give a little bit of an idea of the concept of biocompatibility, which is or is described as the ability of a material to perform with an appropriate host response in a specific application. Whenever we're doing biocompatibility evaluation of medical devices, we're following the guidelines provided by the International Organization for Standardization and also the US Food and Drug Administration, which complement each other more or less. But they kind of give like a baseline for biocompatibility screening of these medical devices. However, these guidelines are very static and they give a very good basis to start this screening, but we need to understand biocompatibility as a very dynamic concept that is constantly changing, it's constantly evolving. As long as we keep on doing research on new constructs, we keep on doing research on how our body may react to those biomaterials. So we need to understand that sometimes those baselines that we have, those guidelines that we have should need to be complemented with other assays or other aspects that might be relevant to understand the response of our body to that biomaterial. And we need to also understand that we should not define biocompatible as safe itself, because something that is biocompatible for a certain application on a certain part of our body doesn't mean that it's going to be, again, biocompatible in another setting or in another application. So these are like very little things that I would like you just to keep in mind. And the ISO and the FDA are going to be described a little bit in more depth by Dr. Sersendo in our next talk, where he's going to talk about in vivo. And so just for you to understand as well, biocompatibility can be assessed at many different levels. It can be described at cell level, tissue, or in vivo in an organism. And today I'm going to be focusing mainly on cell studies, which is also understood as cytocompatibility studies. So what are the things that we could go for when we study cytocompatibility? The main things that we should go for in any biomaterial that we are trying to test out should be viability. Are the cells dead or alive? Metabolic activity, cell proliferation, apoptosis, phenotypic profile. Those are like the main things that we'll go for in any aspect, in any biomaterial. However, as I said before, we should complement all those studies with something that can actually give us a little bit more of an idea of how that biomaterial is being accepted by those cells, such as protein production. Are there certain cytokines or proteins that are we interested in to check out? Cell adhesion, cell migration, ATP production, oxidative stress. I will give some examples of all of this by the end of my presentation so you can have an idea of what we're talking about. And just for you to have an idea, what are the things that we need to be focusing on and keep in mind all the time when we're doing these cytotoxicity studies. The main thing should be the purpose of our biomaterial. So that means, is our biomaterial designed to be a cell carrier? Is it designed to be a drug release system? Is it supposed to have a therapeutic effect on its own? All that is going to define how we create or how we design those experiments afterwards. And also that purpose is going to define as well how we design that biomaterial, which is something that we already talked about on our first talks in the first session. Is it formed by synthetic polymers, by natural polymers? That fact will also define if we use any cross-linking systems, if that is going to end up to a degradable or non-degradable biomaterial. So that is going to affect how we set those experiments. And so you can have an idea, let's say, if we're using a cell carrier, that means that our cells are going to be embedded inside of that biomaterial. If it's a drug release system, that means that probably the cells won't be inside. So we're probably more interested in whatever is going to be released of it, if there are any degradation products of that biomaterial that are going to affect the viability of our cells. So in the first case, we'll be working on a 3D system. So the setting is completely different to the second one, which would be more a 2D system. So the way we plan that experiment is going to vary a little bit. So we're going to see that on the next slides. So before we do any cytocompatibility or biocompatibility studies, we need to understand that previously we should have done a full characterization of that biomaterial in terms of chemical, physical, and biological characterization. And that we also went through on our previous talk. But basically, those are the main things we should understand and fully characterize so we can actually move on to the biocompatibility studies, because that way we could have screened all the characteristics that we want on a biomaterial, and that way we can see what can actually affect or what things we can tackle and maybe change from our design that will also help us in the biocompatibility afterwards. So we also need to understand that the in vitro tests on biomaterials are performed to simulate the biological response of the body when it is in contact with the biomaterial. But again, this is very limited. There are a lot of limitations in vitro studies, as you all know. We will never be able to mimic the exact same situation we have in vivo. Here we're only working with one cell type, for instance, and there are many other factors that can affect the way our body is reacting to that biomaterial. But thanks to this, we can actually have some little hints of how our biomaterial is going to be affected by the direct contact with our body. But again, this should be used as a screening process just to see the main features that we want to keep in our biomaterial. So the thing we need to keep in mind all the time when we're doing all these screening processes is the final use of our biomaterial. And for that, I wanted to put this example of IBD regeneration, which is what we are actually working in this consortium, that one biomaterial that is intended to be applied for IBD regeneration can have many different purposes. It can have mechanical purposes, biological, clinical, and the design of the biomaterial that is targeting each of these purposes will be completely different. So that is what I mean by keeping in mind the final goal of that biomaterial. So what type of cells should we actually work with when we're doing these cytocompatibility studies? Usually at the beginning, what we try to find is a type of cell that we know very well, that it's fully characterized, that we know that they maintain their genetic and morphological features such as fibroblasts, and that have a very good lifespan, and that we can actually see reliable changes and reproducible changes when we're testing it with biomaterials. However, it is good to keep in mind that the closest we are to the in vivo setting, the better. So for instance, if we're working with, let's say, wound healing, maybe working with epithelial cells would be better than working with fibroblasts. Or if we're working with myocardial infarction, maybe working with myocardiocytes would be a better idea. So we need to kind of balance the relevance of our findings with the final application. So now I would like to go through the different main aspects that we will go for when we're testing cytocompatibility. So the first one would be viability. So viability is mainly saying, okay, are the cells dead or they are alive? The gold standard assay, or the main assay that we work on with in our lab, would be the live dead assay. It's a very simple, basically just staining the cells with two dyes, one staining for life, another one staining for dead. The ethidium homodimer stains the DNA of the cells, and it can go inside of the cells because of the permeabilization of the membrane. So it stains the dead cells, whereas the calstain, it stains the live cells thanks to the activity of the esterase of live cells, which turns this calstain to a fluorescent dye. So based on this, here you can see some examples of different biomaterials. The first one that you see on your right-hand side, it's a collagen and HA hydrogel, which has some MSCs encapsulated. And the second one is an HA hydrogel, also with cells encapsulated in them. And here live dead was used to see if cells were alive or dead. And it's very useful because you can see, it allows you to see in situ how your cells are going on, and also distribution within the gel. And the good thing about this is that you don't need any pre-treatment of the sample, any fixation or anything before doing that staining. So that's very nice. However, it has many limitations. If we're working, for instance, with 3D systems, it's very good for 3D systems, but again, it has many limitations according to the type of biomaterial you have. If it's very stiff, for instance, or if it's not very transparent, you might have many difficulties to assess the the viability of your cells by imaging. So maybe other assays will be better, for instance, flow. But again, we will see some examples afterwards, but this is like one of the gold standards that we have in our lab. Then for metabolic activity or cell proliferation, for metabolic activity, there are many types of assays that are have been used for many, many years. But in our lab, we're mainly working with Alamar Blue assay. And Alamar Blue is basically measuring the viability of the cells or metabolic activity by applying this weakly fluorescent compound, which is resveratrol, and you're basically measuring the capacity of your cells to reduce that compound. So the more they reduce, the more metabolically active they are. But that doesn't necessarily translate to healthier cells. Maybe they are more metabolically active because they are inflamed. So that we cannot say, but it is a very, very basic assay that can give us some hint, at least if the cells are still metabolically active. And here we have an example as well of a 3D encapsulated system. And the problem here that we can have as in a 3D system probably is that the stiffness of the gel as well as the porosity may affect that these cells and these compounds can actually diffuse inside of the biomaterial and actually have reproducible readings. So a good solution to this might be, for instance, degrade the gels and just extract the cells that have been embedded and then do a Alamar Blue of cells in suspension. That would be a good idea in case you have these type of problems. And the good thing about Alamar Blue is that it's not a terminal assay. So that means that you can actually assess how the same cells are evolving and are acting at different time points. So you can have a good timeline of how your cells, the same cells, are reacting through time. And usually when we're working with metabolic activity in this type of assays, we should normalize this metabolic activity by DNA content or cell number. And that can also give us some idea of how our cells are behaving through time, if there are any effects on the proliferation of our cells. So if we are normalizing by DNA quantification, we usually use picorin, which is a very simple luminescent assay. And then cell number would be proper counting by DAPI staining. If we're going for apoptosis, for instance, there are many ways we can assess apoptosis, but one of the most powerful tools that we have in any sort of assay would be flow cytometry. And in our case, we're using annexin-5 conjugated with PITC to assess for the apoptotic cells and then propylium iodide for dead cells. And by combining those dyes, we can actually have very nice screening, as you can see here, and quantify how many cells are in early apoptotic stages, late apoptotic stages, alive or dead. And it can give us the quantification right away. It's very simple, it's very easy. And the only thing that we need to take into consideration if we're working with 3D systems, I'm always giving the same example because that's basically what we're working on in this consortium, but if we're working with 3D systems, we need to extract the cells out of that biomaterial. So that could actually add another confounding factor there by the fact that probably you're diminishing the viability of your cells through that extraction process. You don't know that. So that's a previous step you should do before doing flow. And also it works for single cells. So for instance, if we're working with cells that are characterized by forming clusters, this approach wouldn't be suitable. So we can also go for simple stainings of annexin or analyzing gas phase release, like many other ways. And then also phenotypic profiling. Here I just put a simple example. The gold standard way to do it would be qPCR. And here you can see an example of HA hydrogel that was used to reduce the capacity of spinal cord cells to promote neuronal growth. And here you can see that the biomaterial was actually being used to change the phenotypical profile of the cells. And now I would like to give you some examples of specific things that we would like to test and see at certain different applications according to the type of biomaterial. So for instance, if we're interested in looking at the secretome, here it was a biomaterial that was designed to encapsulate MSCs. It was an HA, a collagen hydrogel encapsulating MSCs that was designed to see if it could actually promote angiogenesis. And here you can see that the fact that the cells were encapsulated inside of them, the MSCs were producing a lot more angiogenic factors. So maybe you're interested in seeing if you're actually promoting that aspect. So you would go for ELISAs or proteomics, etc. Cell adhesion would be also very interesting in case that, for instance, you're working with coatings. Some biomaterials are basically coatings that can promote the change on the behavior of several cells. And in this case, it was an ECM coating where cells were applied on top. And maybe you would do SEM just to see if the cells are actually attaching to the surface and see if there are any changes in their morphological characteristics. Or also you would like to see if there are any focal adhesions and if your cells are actually adhering to each other, and you can do stainings, for instance, for integrins or many other ways. But SEM is a very powerful tool and very, very useful. Then for cell migration, usually cell migration is used when we are working with wound healing intended biomaterials. So it's the gold standard assay, and it's very, very simple. It's just a scratch assay where you basically scratch the bottom of your well and you measure at different time points how that area that you see is reducing. So that way you can have a nice timeline on the rate of migration of your own cells. Then we have ATP production. Maybe you're interested in seeing if the metabolism of your cells is being altered by the presence of your biomaterial. And in our lab, we have what it's called Seekers, which is a machine that it's very nice and very powerful as well, where you can see how the respiration, oxidative respiration of your cells is actually being modulated at different time points by the presence of your biomaterial. And lastly, for oxidative stress, here you can see another example of a biomaterial that was a drug carrier that was reducing the oxidative stress of the cells when it was applied to the cells. Just to give you a little bit of a take-home message here, I hope you could see that there is no perfect assay to determine biocompatibility in cells, at least. So the only thing we can do is try to gather as much information as possible in order to understand the possible effect it may have that the application of that biomaterial, and that this is not a definitive assays. So it's just like a process of screening where you can kind of define what are the characteristics that you can actually decide to go for the next phases, which would be the in vivo. And just keep in mind that the design and the aim of the material are key to plan and understand our cytotoxicity experiments. So thank you very much for your attention. And